Welcome back to our session on research priorities for ultra-rare inherited bleeding disorders. I'm really looking forward to this discussion. We know that ultra-rare disorders have very few therapeutic options today and are underrepresented in research. But the urgency of the topic became ever more clear to us in our listening sessions over the last year. It is clear that the community is eager for progress here, and I think that the summit discussions will help to crystallize key areas where we can make a difference. Importantly, we really need to listen and learn directly from people who represent this segment of our community to drive research to what matters most. Before we get started, we've asked Amar Hader to provide a brief overview of her experience living with an ultra-rare bleeding disorder. Amar is a young woman living with severe factor 13 deficiency, along with her 3 other family members. She is currently a student studying graphic design and is actively participating in different art activities throughout the city and is active within her hemophilia chapter, Hemophilia Foundation of Michigan. She was part of a study for a new treatment for factor 13 for 10 years. With new treatment opportunities that recently became available, her quality of life continues to improve. An important aspect of our discussion on research priorities in this area is to find new ways to help therapeutics move through the regulatory process and actually become available outside of clinical trials. And with such small patient populations, we have to think differently about the design and expectations of these trials to ensure the potential treatment has demonstrated safety and efficacy appropriately. To help us understand how regulators are thinking about this rare space, we're thrilled to welcome Dr. Peter Marks, Director of the Center for Biologics Evaluation and Research at the Food and Drug Administration. Dr. Marks is very familiar with the challenges faced in rare disease drug development and has been a champion for designing and implementing innovative strategies that can help us move quickly to improve patient care. Dr. Marks will speak to us about the work underway at the Food and Drug Administration to support this and how clearly defined research priorities for bleeding disorders can help shepherd therapeutic programs. I see tremendous opportunities here to accelerate progress by working together across the community. So let's get right into the discussion.
Hi, my name is Amar Hader. I'm a student studying graphic design at Wayne State University, and I just so happen to also have severe factor 13 deficiency. Ever since I was a few months old being diagnosed, and that leading to my older brother being diagnosed, it's been a wild journey ever since. Me and my family, we weren't very connected to the hemophilia community, being Arab American. We don't typically like to put that out in the open. So in 2017, we decided to be a part of our hemophilia chapter, Hemophilia Foundation of Michigan. And ever since then, it's been a crazy journey of learning about our bleeding disorder and really being immersed in the experience. Unfortunately, because we have an ultra rare bleeding disorder, we don't really fit in with people with hemophilia A and B and von Willebrand's because they have their certain struggles and we have other struggles like miscarriages and infertility issues. And whenever we go to the big conventions, it's always prioritized around hemophilia A and B patients, which makes sense because there's more of them within their community. So I hope that being a part of this program, we can really pay attention to those with ultra rare bleeding disorders, and it can bring more awareness to help with funding and research and listening to our stories as well.
Okay, so basically, I was diagnosed with factor 7 at the age of 15. Living with it, it's been difficult. stressful. It's a emotional roller coaster. Because you have pains here and there. It is something is very difficult to live with, but also I could truly say it blessed me because I didn't think I was gonna be able to have kids, and with the factor 7, I got blessed with 3. And so I call them my 3 miracle babies, and dealing with the virus is just like it is difficult. It's like you have your ups and downs with it. Like some days you'll be okay in the end. Some days you don't because you had a crap in, then you're not at a crap in. And it's a lot. And I'm super glad to do this interview speaking on factor 7 deficiency because I was super afraid to talk to people about it. And now I'm opening up to be open to the world, to let the world know that you're not alone fighting this factor 7, because I know it's very hard and very tough to deal with. So I'm just glad I had this opportunity to share with you guys that you're not alone in this fight with the factor 7, with this bleeding disorder. We is here to come together to lift each other up.
Thanks very much for inviting me to give this talk, and thanks for joining today. Today, I'd like to talk about a regulatory framework for individualized therapies. And this framework is actually quite applicable to rare bleeding disorders. And I'll try to give an example of that today. So I'd like to cover kind of the need, to describe the need for individualized products, talk about challenges in product development, and talk about our efforts at FDA to facilitate product development and overcome some of the challenges in product development. So kind of as a paradigm, even though it's not in the bleeding disorder space, the one approved gene therapy that we have that's administered systemically right now in the United States is anasemnogene. Anasemnogene or Zolgensma is for the treatment of patients less than 2 years of age with spinal muscular atrophy who have confirmed biallelic mutations in the SMN1 gene. This type of spinal muscular atrophy usually presents within the first few months of life. And usually by the time these children are 6 to 12 months old, they're already showing very significant signs of the disorder. They are not able to meet developmental milestones, unable to sit up. And by about 2 to 3 years of age, they're on a ventilator and eventually succumb to the disease. Now, originally there was an RNA therapy developed for this that had to be given repeatedly, but subsequently gene therapy was developed. And that gene therapy, when administered once, was evaluated. The graphic on the left here is one that shows the children who received this. There were 15 children who received this early on in life. The dotted line at the number 40 is if you're below that line, you're abnormal. If you're above that line, you are normal based on this scale. And you can see that 14 out of the 15 children who received this going out over 2 years are now normal, when normally these children would have had a dramatically different outcome. So very impressive results here. The child on the right is shown because she was brought down to Capitol Hill, was running around. Really a remarkable result because that child, based on what we know, should not be, certainly should not be running around and probably would not even be with us without the gene therapy. So this is an example of how successful gene therapy can be. But there are a lot of obstacles to be overcome to achieve this. And that's one of the things we'd want to talk about as we go on today. So, you know, there are a variety of rare disorders and we now know that they number in the thousands, and there are at least hundreds of these that are single gene disorders that could potentially be addressed with current gene therapy technology if we had the ability to do the throughput and the studies necessary. And that's a big if. It's also notable that there are a variety of diseases that are probably a little more common or that actually reduce to very rare diseases when you look at the molecular level at the DNA mutations. And if one were to try to correct DNA mutations, you might actually realize that, even though the disease looks like something that's similar, underlying it are a whole host of different molecular defects at the DNA level. So really important for us to understand how to handle these disorders. And one of the paradigms that we're starting to see here is this transition from what we used to think of as personalized medicine, which was finding the right medicine off the shelf, to individualized medicine, which is creating the right drug to treat the patient. And there are two general types of products that we kind of think of in this space. One is customized products, in which the various products all are for the same indication and have the same mode of action, but that they have something that's somewhat different about them, but there's more in common than is different. And what I mean by that are things like personalized cancer vaccines, or for that matter, chimeric antigen receptor T cells, where although there's something unique about the product, they're really all designed to address the same problem. And then we have created products. Created products have different indications and different modes of actions. And these basically are where one has gene therapies that might be using the same vector, the same thing to carry them in, but different inserts, so that you might address two different bleeding disorder mutations, two different hemoglobin mutations, et cetera. These created products are a little bit more challenging, and these are the ones we'll talk more about today. In some ways, I like to use this analogy that this individualized medicine is moving into this transition from what personalized medicine was really ready to wear. You find something off the shelf that fits you. The customized products are essentially like made to measure products, where you might change the seams around so that they fit very nicely. And these created products are more like bespoke products, where you really are creating them from scratch, but using, for instance, the same cloth. So let's think about factor V deficiency, or parahemophilia. And just for those who aren't intimately familiar, factor V is 330,000 kilodalton glycoprotein that's made in the liver. It's taken up into megakaryocytes, which are the cells of our body that help form platelets, by endocytosis, and then it's present in platelets. These megakaryocytes undergo a very interesting process in which they spin off platelets as they mature. Factor V-A is an integral component of the prothrombinase complex, which is needed for coagulation. And it's a relatively rare clotting factor deficiency, present in about 2 per million, or 1 in 500,000. And there are a variety of mutations that have been described. The interesting thing about this disorder, the manifestations vary from mild to severe. The severe cases, though, can be quite debilitating. And there is no factor concentrate per se. One gives plasma to deal with this disorder. So one could imagine that using genome editing technology for certain mutations here, you could potentially use the advantages of genome editing technology to fix a single base pair mutation that's causing factor V deficiency. Genome editing technology allows one to have DNA to be inserted, deleted, or replaced in the genome of an organism using these engineered nucleases or molecular scissors. The one that we often hear about is CRISPR-Cas9, which is one of several different genome editing tools. But what has happened over the past year since CRISPR-Cas9 was first described in 2012 has been the development of successor products, which are now capable of doing essentially single base pair editing using a modified CRISPR-Cas9 construct. So you can now actually convert a given abnormal base pair in the DNA and ultimately then repair a single base pair defect. So you could potentially think about applying this in this type of a disease to fix this. It's a very large protein, so replacing it with a gene therapy gets to be messy. And so fixing it might be more a more forthwith way to do this. So if you were to do this, a hypothetical therapeutic concept would be to correct certain causes of this defect, which are pretty rare. So this is going to be something that's going to be a therapy for perhaps at most dozens of people around the globe. But you could correct one of the missense mutations. That's when one base pair is messed up. And then correct that so that you have a normal functioning factor V gene. And you to do that using this modified CRISPR-Cas9 enzyme in this particular case to do this, and you would basically express that in an AAV8 vector. Now, so what if we wanted to do that? What are the challenges that would exist in implementing this concept? So the challenges of such individualized gene therapies come in various flavors. They are in manufacturing, which is probably one of the most significant. There's also ones in non-clinical development, clinical development, and then in product access. And let me take you through those challenges, and then I'll tell you a few of the things we're trying to do to address those challenges. So for manufacturing, in gene therapy today, we have a very awkward situation, which is that commercial manufacturing to develop a process requires one to manufacture enough of that gene therapy to recoup the investment in developing the manufacturing process, such that you need to treat populations of 100 or 200 people for a gene therapy to be of commercial interest. Much larger than that, a couple of hundred to maybe 1,000 people a year for a systemically administered gene therapy, that we don't have the technology to do that yet because of the cell lines, et cetera. That'll come with time, probably. On the other hand, for the very small populations, we can do that in kind of laboratory scale, but we don't have really robust commercial processes to do that in a cost-effective manner right now. And so we need to solve that because many of the gene therapies we're talking about for these very rare disorders, one might need 20 or 30 doses a year. And so one needs to find a way forward to be able to make these in a cost-effective manner and in one that is sustainable over time. So there are some ways we've been thinking about this. And one of the ways that one can help when one needs to do something over and over again is one uses a device. And this is not a gene therapy machine. This is a soda machine. I know that. But the idea here is it is an example of what you might imagine could happen, because a fair number of the gene therapy manufacturing pieces are the same. You have to grow up the vector. You then have to essentially purify the virus from the cells that grow it up. And then you have to clean up that preparation so that it's acceptable and can be formulated to give to someone. Those downstream steps can largely be automated, and potentially even the upstream steps can be automated. And so you could imagine that it might not look exactly like this, but you could imagine ultimately 5 or 10 years down the road, you could have a machine that could make a variety of different gene therapies so that you could do this much more efficiently. Hypothetical, but possible. And that's a potential solution there. The other issue in the non-clinical development, which is basically showing that these things work in proof of concept, which is important because you'd like to have animal models or at least cell line models that help show you that these things are going to work, since you're not going to be using them in large numbers of people. And right now, our problem is that animal models may be less than ideal. One of the problems is that CRISPR-Cas9 is a human genome editing construct. The way this thing works is there is a segment of this complex, which is RNA, that matches up to the DNA. And unfortunately, it has to be human to really figure out whether we have off-target effects, that is, whether it's matching up in the wrong place. And so we need to find models which allow us to look at human DNA in the preclinical setting. So there are ways now that we can do that. Some of those include using some things called human organoids, which is what you can do is you can actually make structures in a petri dish that resemble human organs. It's been done very well for liver, but it's also now being done for kidney and other organs as well, even the brain. We can also use something called humanized mice, where the livers of the mice essentially can be made to be very similar to a human's mouse, human's liver, and that can help facilitate safety testing. For clinical development, the question is, how do you appropriately document the natural history of disease or collect baseline data? We're lucky in that for many of the bleeding disorders, as opposed to some of the neurologic disorders, we actually have reasonably good natural history data, at least for some families, and that can be very helpful. But ultimately, determining efficacy in very small populations can be challenging. Now, again, in bleeding disorders, we're a little bit lucky because for at least for some of the disorders, we know the approximate factor levels that are associated with normalization of a bleeding tendency. So that's helpful here. But even so, you're going to have small populations that you're going to treat, and you need to figure out how to make the most of every patient. And so one way of doing that is by using novel clinical trial design. So we're trying to figure out ways to address this small population question by figuring out how to help people collect baseline data in rare populations. That is, how are people doing before they're treated, so that when you come along with a treatment, they serve those people who you've studied can serve as their own controls. And the other thing is to use Bayesian clinical trial designs where every patient counts. So the first patient who's treated, you don't know for sure whether they're going to respond or not. If they don't respond, well, the next patient, they still could respond, but the likelihood might be lower. On the other hand, if the first patient responds wonderfully, well, then the chance of the next patient responding is somewhat higher. Not 100%, of course, but with each patient you put through, that percentage can be recalculated, and the likelihood eventually probably crosses some threshold that says this is a viable therapy. And then there's this issue of product access. And this is obviously incredibly important because we can do everything we want about producing a therapy, studying it, but if people can't get access to the therapies to help them, then it's all for naught. And so we need to figure out how to produce therapies and to continue to produce them in a manner that's not cost prohibitive. So for the short term, we're thinking about public-private partnerships to get to product access, to allow diverse populations to get products through a kind of a streamlined production process. We have kind of currently through the National Institutes of Health and the collaboration that we have with them and the Foundation for the National Institutes of Health, which is a nonprofit group that supports the National Institutes of Health. We are standing up a bespoke gene therapy consortium, which will allow, hopefully, us to start to develop these products and move them from gene therapy constructs into products that can potentially help patients and then be produced on an ongoing basis as needed. The way we're going to try to get there through the various stages is by starting to simplify some of the steps. So what are we talking about here? Well, we're trying to develop some concepts here that should hopefully make things easier. Right now, one of the challenges we have in rare disease gene therapy development is that very often the individual who develops a gene therapy for a rare disease is not necessarily an expert in gene therapy development. Oftentimes, it's the physician or healthcare provider who has been caring for patients with a rare disorder. And so they find someone that can help them develop the gene therapy, which means that we have a lot of variation in the types of gene therapies that are developed. And that includes with manufacturing processes and characterization processes of the ultimate product. The idea here is if we can put together a cookbook for the development and manufacturing of bespoke therapeutics, we could hopefully eliminate some of that variability, ultimately add value to these products, because if you came to a commercial entity with something that the commercial entity could rely upon as being something that they could transfer into their manufacturing process, as opposed to have to re-engineer the whole process, there would be greater value there. Additionally, we're trying to look at ways that we could streamline product development by taking an advantage of a fact of gene therapies, which is that the gene therapy vector, it could be AAV9, adeno-associated virus 9, or adeno-associated virus 8, or some other gene therapy vector, that the backbone, when you have it, as long as you put in the inserts of an appropriate size, it functions a lot like the razor handle when you have different razor blades. And so it's functioning in kind of a little bit of like a device. And so the idea would be to try to leverage as much information about that vector from product to product. So we've come up with this concept of an originator product, which would be a well-characterized product, which you've very well characterized both the vector and the insert, and then offshoot products, which use the same vector, but different inserts. And that would hopefully allow us to leverage information on file and focus on the distinguishing attributes of offshoot products. So just kind of to reiterate this, for those of us who are old enough, we got our start, some of us got our starts in the laboratory doing molecular biology using a Maniatis set of kind of do-it-yourself manuals. And the idea here is to try to develop something very similar for gene therapy. And for these leveraging information from one application to another, the idea is that you have a well-characterized originator product, which serves as kind of a reference. And that originator then can be used to have multiple offshoots, which use the same manufacturing process, the same purification process, the same formulation process, and presumably, with the exception of having to look at any unique toxicology characteristics related to the gene insert, the vector would have the same toxicology from product to product, allowing one to leverage a lot of the information on file. So when one went to do regulatory submissions, you could really streamline the process. So really here, we're trying to advance the development of cell and gene therapies for populations of all sizes, working to overcome limitations in manufacturing, trying to individualize product development, and sometimes that means for very small populations. We're working with sponsors to provide input and to collaborate on novel endpoints, and we're really encouraging innovative clinical trial designs. Ultimately, the goal here is to try to expedite this field so that rather than the relatively slower pace that we've seen going from gene therapy to gene therapy over the past years, we can see this really accelerate moving forward. So thanks so much for listening today, and look forward to the continued conference.
Thank you for joining us for a brief summary of the National Hemophilia Foundation's State of the Science Working Group 3, Ultra-Rare Disorders. My name is Dr. Amy Shapiro, and I am from the Indiana Hemophilia and Thrombosis Center in Indianapolis, Indiana. My co-chair in this working group is Dr. Diane Nugent, who is Chief of Hematology at CHOC UC Irvine and the Center of Inherited Blood Disorders. Dr. Suchitra Acharya, who is Director of Hemostasis and Thrombosis at Northwell Health and Professor of Pediatrics at Zucker School of Medicine at Hofstra Northwell, also led a subgroup. Both Doctors Nugent and Acharya will present a high-level overview of their subgroups. The inception of the State of the Science endeavor began with NHF conducting listening sessions to align focus between stakeholders, to define priorities for future research, and to foster progress through collaboration. The initial research priorities identified through the listening groups for ultra-rare disorders included targeted and accessible diagnostics, therapy that is inclusive of all disorders and phenotypes, applicability and potential for gene therapy, underlying biology of ultra-rare disorders, many of which have been poorly studied as compared to factor 8 or 9 deficiencies, data collection across the lifespan, including issues related to aging, and patient-reported measures and overall outcomes. Important to all involved in ultra-rare inherited bleeding disorders, including patients, families, and care providers, is that now these often overlooked conditions have been identified as a focus area from a national perspective. By doing this, we can highlight and emphasize the needs of these underserved populations and the gaps in knowledge and care, with the goals to advance care and improve outcomes. This process included the development of expert panels representing clinicians, researchers, laboratory experts, industry representatives, community members, multidisciplinary hemophilia treatment center members, and representatives of NHF to address each specific topic to identify priorities. Our working group on ultra-rare inherited bleeding disorders worked across diagnoses to advance care for all. Our working group divided our ultra-rare inherited bleeding disorders into 3 focus areas or subgroups, with experts to address current gaps and help chart a path forward. The categories we identified included 1, diagnostic, systems biology, and mechanistic science, led by Dr. Diane Nugent. 2, clinical data collection, research infrastructure, led by Dr. Suchitra Acharya. And 3, regulatory processes for novel therapeutics and required data collection, which I led. These 3 subgroups are listed here with their members. Each subgroup included a diverse range of experts that contributed their knowledge, perspective, and insights. I will now turn this presentation over to Dr. Nugent to present a summary of the subgroup on diagnostic, systems biology, and mechanistic science. Following Dr. Nugent's summary, Dr. Acharya will provide a summary of the subgroup that addressed clinical data collection and research infrastructure. I will then present the subgroup on regulatory processes for novel therapeutics and required data collection.
Thanks, Amy. For subgroup A, we've really focused on diagnostics, systems biology, and the needs of mechanistic science. Why is this important for our patients? Well, clearly, our patients have gone through what's known as the diagnostic odyssey, especially the ultra-rare patients, often seen by their primary care physician, who may recognize that there's a bleeding problem. The patients then get bumped around to various centers, and the more rare and complicated the bleeding disorder, probably the less likely is that an appropriate or specific diagnosis is made. And we want to understand these rare disorders better because it really helps give us insight into the normal functioning of the coagulation system. So for all of these reasons, the Diagnosis and Research Committee was formed. I am the lead of this section, but truly every single person contributed equally, if not more than I did. We had really robust discussions. And as you can see from this list, truly major leaders in the area of research of coagulation, the genomics of coagulation, and diagnostic services. And again, just can't really call out one person over another. It was a really dynamic group with a wealth of experience. So very fortunate to have these individuals volunteer so much of their time to this cause. So what were the key issues that we felt were important for advancing the needs of patients with ultra-rare disorders? Well, as I referred in the beginning, we really need to create a roadmap from the community physician who first identifies a patient with a bleeding disorder and get them to centers where a diagnosis can be made, and then appropriate referral to an HTC or a center that understands how to treat, how to prevent bleeding, and to really provide optimal care for patients so that they can live a full and really meaningful, productive life. So how does this roadmap or journey from the community physician to an area of diagnosis occur? Many times an MD will do the routine, well-known screening labs or DIC screen, but often the screening lab is not diagnostic. And then that sort of is where the first stall occurs. So how does a community physician know where to send samples or how to find help? We suggest and strongly believe, hypothesize that a telehealth access or e-consult to a national center for information and help in further workup is needed. That can be centered or promoted by a national organization like NHF or the HTC network through HRSA and 340B funds. A website that is easily accessible by community physicians to know where diagnostic centers are and where a patient can be referred are really key. And core diagnostic labs where more advanced studies in what might be called research into an individual patient really need to be supported. And so this national network for advanced diagnosis would include centers usually at large institutes, university centers, sites that are really devoted to the research and specialists in coagulation. Many times there are both a combination of physician scientists and laboratory postdocs and scientists in coagulations at these centers. Then a national database, I'm sure that this has come up in almost every center, where we can collect phenotypic and family history, strong linkages with the HTC networks, both for referral information and continued workup and evaluation of patients and families as needed. We need to train the workforce at the HTCs and referral centers on what the options are and how to obtain and process samples properly so that the pre-analytic factors don't confuse or negate a diagnosis. And we really need to create policies to allow for patient samples to be sent for investigational studies without having to do a unique IRB for each individual patient. When patients are only 1 in 2 million, we're not going to have a whole lot of support for an IRB form for that. So beside the national centers for diagnosis, we also need national research centers. How do researchers find the patients of interest? How do MDs find the researchers? We need to have a mechanism for matching up referring physicians, even HTCs, to sites that can do the kind of research that's needed on many of these rare disorder patients. However, currently funded researchers really can't use their own NIH monies to do this work, which sometimes can take years. to get to a final diagnosis or gene variant. Challenges also begin to appear with diagnosis when it is outside of CAP/CLIA laboratories. We truly need hybrid labs that can both do investigational research and report out that research in a CAP/CLIA scenario. And this would be a national innovation. This is something new that doesn't exist at present, but we really need to work on. And I know that there are many people on our team that would be willing to try and define what that would look like. Workforce, of course, also needs to be trained to look for rare disorders and how to work them up and approve at these advanced diagnostic centers in CAP/CLIA labs. Finally, the genomic approaches, which many of us put our hope in, there's still cost as an issue that prevents patients and payers from getting to a diagnosis. And although we talk about how the cost of whole genome or whole exome is going down, when it comes to an individual patient trying to find access to those centers, it's still a very daunting course with many, many challenges. And I can speak to that every day of the week that we hold a clinic. We have challenges with whole genome because there are way too many variants of unknown significance at present. Even doing family studies, this has not helped us a whole lot, but we're getting there. But just doing whole genome usually does not make a diagnosis in most cases. And we're getting more effective or better hits on really focused next-gen panels, both for platelets and clotting protein factors. Very few commercial companies are interested in focusing on rare disorders, so that's a challenge as far as access to diagnosis through a genomic approach. And ClinVar, which lists all the variants that are associated with disease, it's growing, but there's still large phenotype, genotype data that needs to be entered and linked. And again, not necessarily focused on bleeding disorders. So we really need to build our workforce there. The work that's done by My Life, Our Future has been really helpful in moving this along. Only about 50 to 60% of unknown bleeding disorders are diagnosed, as I said, with whole genome, but we're getting better. It's important to realize, though, that even with the best screens, there are disorders that are associated with post-translational changes or processing defects, splice site variants. So we have a lot to learn and a long way to go with genomic studies. And finally, there are patients who are misdiagnosed as having an inherited defect that are actually acquired bleeding disorders. And we need to be able to do a better job identifying and helping with those. So key things, linking the community physician, helping the journey to diagnosis for patients, identifying sites that can merge research and diagnosis together, and really expanding and supporting genomic approaches with large data banks is really key. And we have a lot more details on that, but I'll hand it over to the next speaker. Thank you very much.
Hello, everyone. Thank you, NHF, Len and Michelle and Donna for this opportunity, prioritizing the needs for ultra-rare inherited bleeding disorders, working towards a national blueprint for the same. Our subgroup for working group 3 was entrusted with priorities for clinical needs, data collection and research infrastructure. And I'm doing this presentation, presenting a summary of our discussions on behalf of all my colleagues who are listed on the slide. And I want to thank each and every one of them for their robust discussions along with Amy and Diane. So one of the first clinical questions we wanted to answer, and these questions came from NHF. Do defined severity categories exist to predict outcomes and guide treatment? We are all aware that the ISTH bleeding assessment tool has been validated for von Willebrand's disease and some of the platelet function defects. We need to validate this for the ultra-rare bleeding disorders. And once we do that, it might be helpful to define mild, moderate, and severe phenotypes. The European Network of Rare Bleeding Disorders already has a severity classification, so it would be helpful to compare it to that classification to see how similar or dissimilar our classification is. And also, if possible, to highlight specific phenotypes for individual ultra-rare disorders. For example, umbilical cord bleeding is pathognomonic for factor 13 and fibrinogen disorders. So maybe there are other disorders also have something similar. And eventually this will help us make an accurate diagnosis of these ultra-rare disorders to prevent misdiagnosis and mismanagement of these patients. Do treatments exist to address clinical manifestations and prevent sequelae, global versus specific treatments? We discussed using precision medicine and tailoring and modeling out of the Children's Oncology Group model, where a particular question, a treatment question typically is asked in the pediatric cancer patients, and a protocol is devised, and that's how they've made a lot of progress in the management of cancers, childhood cancers in these days. I think it would be important for us as a group to come up with some algorithms for the use of single factor concentrates alone in combination with antifibrinolytics or antifibrinolytics alone, depending upon the type of bleed or depending upon the type of procedure these ultra-rare patients need to undergo. And would also be important to come up with guidelines for prophylaxis. I'm sure is a subgroup who would definitely benefit with prophylaxis, and it would be nice to have guidelines for the same. Currently, I would say a lot of these ultra-rare patients do seek care at national hemophilia treatment centers, but there are also a few out there in the non-HTC settings. And so I think coming up with a system to capture those non-HTC ultra-rare patients would also be important. What are quality of life impacts, and are they collected? Currently, I don't think we have anything designed for the ultra-rare bleeding disorders. ATHEN Transcends is a natural history study using safety as an endpoint, and aims to collect good quality data as part of good clinical practice. And ATHEN Transcends also has these quality of life questionnaires as listed here for hemophilia. I think these could potentially be modified for the ultra-rare bleeding disorders. Do we have centralized data collection capability to define natural history treatments and outcomes? Again, currently what we have is the ATHEN RBD data form in the form of electronic CRFs entered into Clinical Manager, which is being modified to mimic REDCap in order to maintain privacy. It would be important to collect natural history of these ultra-rare bleeding disorders from birth to aging adult, to actually evaluate risk versus benefits of products that we use, both single factor concentrates, antifibrinolytics, and other products. Also to evaluate prophylaxis versus demand therapy, what are the benefits of that? Whether certain bleeds, certain procedures require only an antifibrinolytic versus using replacement products. For those ultra-rare disorders for which there is no single factor concentrate available, whether there's a role for non-factor products, and are they safe? Assessing, again, quality of life throughout the lifespan. Collecting data through ATHEN in a single database would also give access to patients for clinical trials, especially since these are such rare disorders and not one center or even a nation has enough patients to be able to do this. And then harmonizing the US database with the European Union and other global databases. This will also help satisfy regulatory requirements needed to approve products quicker. In terms of the research infrastructure, I think the biggest current impediment is for providers needing resources to enter quality data into Athens. So definitely that should be a top priority. We also need a centralized lab to do antigen activity levels for these ultra rare disorders, also to do genotyping, and also to look at modifier genes, epigenetics, which may have a role in modifying the phenotype. And in order for this to happen, we need to standardize the antigen activity measurements using and doing quality control so that once we have a central lab, institutions can actually contract with this central lab and overcome insurance hurdles, which we always face to make an accurate diagnosis of these disorders. Athens currently has a repository through Varsity, which has started banking samples, but I think it would be important to do serial sample banking throughout the lifespan in order to better understand these disorders. Athens Transcends can also give researchers with funding access to clinical data as well as clinical data so that they can look further into modifying genes, other epigenetics, to see if there is even a genotype-phenotype correlation. Athens Biorepository will need to be connected to next-gen sequencing and labs which do study modifying genes in this particular population. And lastly, I want to put forward, in a utopian world, it would be great to have a central lab to confirm the diagnosis, do genotyping, look at other modifying genes, access to serially banked samples, so that we have an opportunity to make an accurate diagnosis of these ultra-rare disorders, and also possibly identify bleeding of unknown causes. And in this manner, we'll be able to reduce costs surrounding diagnosis and treatment, as well as reduce the inappropriate use of currently available products. Our group also came up with another great suggestion, that is establishing a utilization management group, like a triage center, where if you suspect a rare bleeding disorder, you could go on the website, order specific tests, get some expert clinical consultations, as well as genetics consultation to interpret the significance of genetic testing results and ultimately make an accurate diagnosis. Thank you.
I wish to thank all members of our subgroup for their thoughtful participation, effort, and energy that was clearly evident throughout our meetings. The time that each member dedicated to our work was outside their current responsibilities, added onto very busy schedules, and demonstrates their personal commitment to advance efforts for ultra-rare inherited bleeding disorders. We began our regulatory subgroup work by listing current obstacles and gaps for ultra-rare disorders. High-level issues included current regulatory pathways that are geared towards more prevalent conditions and the lack of harmonization between regulatory bodies throughout the world. These issues add burdens of complexity, time, and ultimately cost to the development of therapeutics aimed at very small populations. The FDA is aware that ultra-rare disorders require a more streamlined approach to address very small populations who currently have limited or inadequate therapeutic options. The group discussed the importance of product safety and potential benefit as critical outcomes, rather than proving superiority to standard of care. Many ultra-rare inherited bleeding disorders have inadequate data on the natural history and outcome, and use of currently utilized interventions if these exist. This data is essential to support the need for new therapeutics and their potential benefit. To achieve this, centralized data collection through a national system is required. Through such a system, we can collect needed data to improve knowledge of natural history, patient-reported outcome measures, current therapeutics utilized and their outcomes, and disease sequelae, including the impact of aging. A nationalized data collection system that includes ultra-rare inherited bleeding disorders fosters ongoing clinical learning and ultimately translates into advancement in care. A major issue that confronts biopharmaceutical companies is the need to obtain a return on very large investments to develop and license a new therapy. The potential mechanisms to incentivize pharmaceutical companies to develop therapies should be clearly delineated and, if feasible, increased. Once therapeutics are available, access to all must be assured and reimbursed, including off-label use of applicable agents. Patients may suffer from an inability to obtain needed treatment if such agents are excluded from coverage. The issues discussed in our subgroup were deemed of sufficient importance that our group decided to develop a white paper as a call to action. Although the regulatory subgroups identified priorities will be included in the manuscript that summarize all three subgroups' work for inherited ultra-rare bleeding disorders, this separate white paper will bring a laser focus to a critical component: the path to development, proof of safety and efficacy, and hopefully approval and availability of needed therapies. A high-level content for this white paper has been developed and includes issues related to population sizes, heterogeneity of manifestations, current treatment or lack thereof, need for centralized information on natural history and outcomes across the lifespan, and the need for a medical home. We have outlined the unmet needs that are common across diagnoses and the barriers to optimal care and outcomes. We have proposed potential pathways and concepts to address these issues. The critical questions addressed by each subgroup are listed here. Dr. Nugent discussed the need to improve diagnostic testing with national access. Centralized national testing to include genomics, along with other testing modalities, is needed to associate and delineate disorder-specific phenotypes. National sample banking is needed to advance knowledge as testing capabilities progress over time. Dr. Acharya reviewed the need for easily accessible information for care providers to identify phenotypes and testing for patients requiring diagnoses. She reviewed the need for centralized data collection to support the natural history knowledge specific to each disorder and current treatments and outcomes. When further data is required to support provisional or initial approvals, centralized data is needed to support ongoing learning to advance care. It is important to assure that the national system utilized fulfills post-approval regulatory requirements. Resources to support conduct of research in ultra-rare disorders is needed. Two examples include the ability to enroll participants through a decentralized mechanism, if required, as well as the use of a central national IRB. I reviewed the concerns related to current regulatory processes, including consideration for streamlining pathways for very small populations, especially when limited or no therapeutics are available. The ability to achieve provisional approval based upon commitment to collect post-approval data to support safety and efficacy, the need to incentivize product development, and the need to obtain approval for off-label use of available therapeutics when they are applicable and efficacious. Important issues common to ultra-rare inherited coagulation disorders have been identified and vetted in a national forum by a wide group of experts. It was evident that we were all energized by shared goals. We have identified potential strategies to advance care. We now need to maintain momentum to translate these concepts into progress. We hope to look beyond present circumstances to imagine and work towards a brighter future. Thank you.